Welcome everyone to the Biochemistry series. In this series, first of all, we are going to go through biomolecules. The first biomolecule that we're going to understand is protein. Biomolecules are carbon-based organic compounds that are produced by a living organism. More than 25 naturally occurring chemical elements are found in biomolecules, but these biomolecules consist mainly of carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. In terms of percentage of total number of atoms, hydrogen, oxygen, nitrogen, and carbon together makes up over 99% of the mass of most cells. Biomolecules are mainly classified into two, micromolecules and macromolecules. This classification is mainly based on their molecular weight. So, the molecules which are small or of low molecular weight, they are known as the micromolecules, and those have higher molecular weights are known as macromolecules. Micromolecules mainly composed of sugars, fatty acids, amino acids, nucleotides, vitamins, hormones, neurotransmitters, primary and secondary metabolites. The sugars, fatty acids, amino acids, and nucleotides, they composed of four major families of small biomolecules or the micromolecules. In case of macromolecules, they are the biomolecules which have a higher molecular weight, and they are mostly the polymers of the micromolecules. They include proteins, carbohydrates, and nucleic acid. In today's session, we're going to understand about the basic idea of amino acids, their optical properties, and absolute configuration. Amino acids. Amino acids are the compounds that mainly composed of carbon, nitrogen, hydrogen, and oxygen, and their savers are the monomers or the building blocks of proteins. As the name implies, this particular compound has an amino group attached to a carbon and also a carboxyl group attached to a carbon. So if the amino group and the carboxyl group are attached to the same carbon, then this particular carbon is known as the alpha carbon, and this is said to be the basic structure of an alpha amino acid. The various alpha amino acids differ with respect to the side chain or the R group attached to their alpha carbon. So this is the general structure of an alpha amino acid. The only variation or the only difference between different alpha amino acid is the side chain that attaches to this particular section of this amino acid. Next, about the amphiprotic property of an amino acid. An amphiprotic molecule or an ion can both accept as well as donate protons, which means they can act as both as an acid as well as a base. So if we analyze, the ionization state of an amino acid varies with pH. At physiological pH, that is approximately 7.4, the alpha amino groups are protonated, which means they are an NH3 plus condition, and the alpha carboxyl group are deprotonated, that is COO minus. And how is it possible? Because this particular amino acid has amphiprotic property that they can accept protons as well as donate protons. So at the physiological pH, alpha amino group are protonated and alpha carboxyl group are deprotonated, which means there is a charge separation in here, that is NH3 plus in here and CO minus in here. So the net charge will be 0. So this double ion form, this particular amino acid in a physiological pH, we can say that the amino acids are in Switter ionic form, or Switter ionic form means the double ionic form, that they have charge separation. As I mentioned earlier, the ionization state of an amino acid varies with pH. Let's look into that. In a low pH condition, that is a strongly acidic medium, the carboxylic group is protonated and uncharged. Protonated means it accepted a proton or the H plus. Because it is acidic medium, in that particular medium the amount of H plus ions are higher. So the carboxylic group is protonated and uncharged, which means it received a proton and it is uncharged. The net charge is 0 in case of carboxylic group. But in case of amino group, it is protonated and it is charged, because by the acceptance of another proton, the charge of the particular amino acid become positive. So at a low pH, we can see the net charge of the particular amino acid is positive charge. So overall charge of an amino acid in a low pH medium or a strongly acidic medium is positive. But at a high pH, that is a strongly basic condition, we know that in case of a basic condition, the amount of H plus ions or the protons are comparatively less in case of the medium. So the carboxylic group is deprotonated and negatively charged. Deprotonation means the carboxylic group gave away their protons. So it has a net negative charge. But the amino group is deprotonated but is uncharged because it already gave away the exceptional positive charge which they had. So the overall charge will be negative. So in a strongly acidic medium, the overall charge is positive, and a strongly basic medium, the overall charge is negative. So at a physiological pH, that is approximately 7.4, the amino acids stay as sweeter ions, or in intermediate pH, we can say their net charge is 0. That is why we say amino acids are amphiprotic molecules, because they can both accept protons which act as a basis, and they can donate protons and act as acids. Next, about their optical property. As we all know, all amino acids except glycine are optically active. When will any amino acid become optically active? Optically active means they can rotate the plane polarized light. But what causes this rotation of plane polarized light? Because they possess the property of chirality. Chirality means the possession of an asymmetric carbon. The presence of an asymmetric carbon. Asymmetric carbon means a carbon whose 4 valencies are fulfilled by different groups. Not a single pair is having the same functional group. So, if the 4 valencies of a particular carbon is satisfied by 4 different molecules, 4 different functional groups, then we can say that particular carbon is a chiral carbon or an asymmetric carbon, and this particular one possesses the property of chirality. Molecules that are not chiral are said to be achiral, or without chirality, they don't possess the property of optical rotation, or they don't possess optical property, which means they are optically inactive. So, all amino acids except glycine are optically active, means in case of glycine, there is no chiral carbon. An optically active compound can rotate a plane polarized light either clockwise or counterclockwise. So the optically active compounds that rotates the plane polarized light to the right, they are known as the dextrorotatory, and it is designated by either a plus sign or by the small letter d. And the optically active compounds that rotate the plane polarized light to the left, they are called the levorotatory, which are designated by a minus symbol or by the small letter L. And this optical property is usually measured using polarimeter. Next one is optical rotation. Optical rotation is the quantitative measure of the rotation of light caused by the compound, which means the magnitude represents the extent to which the plane of linearly polarized light is rotated, and the sign represents the direction. Suppose if it says it is dextrorotatory, means that particular light has been rotated to the right, and if it is levorotatory, then the particular light has been rotated to the left side, and the particular value of that particular rotation or the magnitude which represent to which extent, the degree to which the particular rotation happened. The optical rotation depends on many factors. The first one is the concentration of the compound, means the concentration of amino acid that we have taken, the temperature at which it is happening, and the wavelength of light used, the wavelength of the plane polarized light which has been rotated, and the solvent we used to dissolve the sample of amino acids, and finally the light path length. Next, about specific rotation. It is the reference value of optical rotation for a given concentration of compound at a given temperature and fixed wavelength. Suppose we are providing a particular light with a fixed wavelength and the temperature is also fixed, then how much rotation happened, that reference value is said to be the specific rotation, and is the observed value of optical rotation when a particular plane polarized light passes through a sample with a path length of 1 decameter and a sample of concentration of 1 gram per millimeter, which means the amino acid concentration is 1 gram per millimeter and the path length is 1 decameter. So how much rotation happened, that particular rotation, the magnitude of that rotation in 1 decameter path length, and the concentration of the sample is 1 gram per milliliter, that particular value is said to be the specific rotation. Last session is about absolute configuration. All alpha amino acids except glycine have an asymmetric alpha carbon atom and a chiral molecules. We have discussed it earlier. So a chiral amino acid can exist in two configurations that are non-superimposable mirror image of each other. These two configurations are called enantiomers. An enantiomer is identified by its absolute configuration. So different systems are being developed to specify the absolute configuration of a chiral molecule. The common systems that we use in case of amino acid is the DL system and the RS system. We are going to discuss them in detail. In DL system, it is actually a comparison. Here we relate the structure of a 3-carbon aldol sugar glyceraldehyde to that of the amino acid. These are the enantiomers of glyceraldehyde. If the particular hydroxyl group is on the left side of the Fischer projection, then this is said to be L-glyceraldehyde. And if the hydroxyl group is on the right on the Fischer projection, then it is D-glyceraldehyde. Similarly, if the amino group is on the left side, then we said it to be as the L amino acid, and if the amino group is on the right side, it is a D amino acid. But this DL system is for compounds with one chiral center, which means these are applicable only to amino acid with only one asymmetric carbon. Next is about next system is the RS system, which is more advanced than DL system because it is applicable to compounds with more than one chiral center. And this particular system does not need any reference molecules such as glyceraldehyde in here. So it is applicable to almost all the amino acids. Here we use the priority rule. The priority is based on atomic number, which means higher the atomic number, higher the priority. So this is the priority order. In case of SH, the first compound is sulfur and the atomic number is 16. And in case of OH, the first atomic number of the oxygen is 8. So comparatively, SH is given more priority than oxygen. And NH2, of course, N has the atomic number 7. And in case of these compounds, the first particular one is carbon. So what we do is we add the atomic number, and those have the higher value is given the priority. So in case of COOH, that is C is 6, oxygen 8, 8 and 1. So overall the total number or the value is higher than CH3. So it is given higher priority than CH3 and then H. This is the priority rule. So in RS system, first of all, we will align the compounds according to their priority, or we are marking the priority. See here in case of alanine, this is a basic structure of alanine, NH3. So the first priority is given to NH3 because the first one is N, that is 7, and here it is COO minus and a CH3. We know that according to the priority rule, NH3 is having the first priority and CO minus is having the second priority, and the CH3 or the methyl group is having the third priority, and finally it is H. And after numbering this, we find the direction, that is 1, 2, 3. So this rotates in a clockwise direction. So this has a configuration of R. So this is called the R alanine. Here in case of, if the molecule is represented like this, the first amino group is here, carboxyl group is here, methyl group is here, and the hydrogen is here. So the rotation will be like 1, 2, 3, 4. So it will be like anti-clockwise, so the counter-clockwise rotation. So this is said to be S-alanine. So this RS system actually varies according to the representation. So first of all, we will provide the priority order, and then we will follow the direction of the majority of the groups, and then we will assign R and S respectively. That is all about the first session. We will meet again in the next session. Thank you.